Good day. My name is KUYE ADESEGUN Jacobs. I'm from Group 1, 5th course, 6th faculty. I am presenting a webinar on clinical significance of advanced paternal age as a risk factor for de novo mutations in offspring. Special thanks to my class teacher, Dr. Elizavia Mikarikovic. What are de novo genetic mutations? De novo genetic mutations are genetic alterations that is present for the first time in one family member as a result of a variant or mutation in the germ cell, egg, or sperm of one of the parents, or a variant that arises in the fertilized egg itself during early embryogenesis. And one of the major causes of this kind of mutation is advanced paternal age, which has been shown to increase the frequency of congenital diseases such as heart malformation, oral, palate, and lip cleft. And recent research have discovered that these de novo mutations can also cause mental disorders such as autism spectrum disorders, schizophrenia, bipolar disorder, low IQ, as well as ADHD, which is attention deficient hyperactive disorders. We should also understand that this mutation occurs as a result of mutagenesis that occurs in parental gamete during oogenesis and spermatogenesis. Various research has shown from more than 40 million births of babies born to fathers of an advanced paternal age, which roughly equates to older men or older fathers than 35, were at higher risk of adverse birth outcomes such as low birth weight, seizures, and need for ventilation immediately after birth. And according to latest findings, which we were able to establish that there are certain associations between advanced paternal age and offspring risk for psychiatric and developmental disorders. It was said that de novo single nucleotide variants, which are the DNSNVs, accumulate with increasing paternal age. However, the actual magnitude of this risk from mutations in the male germline is practically or theoretically unknown. So major findings upon investigation interpret whether epidemiological association between each disorder and older fatherhood are consistent with the estimated role of this DNSV, which are the de novo's single nucleotide variants. However, it was shown that paternal age-related de novo mutations confers a small amount of risk for these disorders. And according to various reports for autism spectrum disorders and schizophrenia, epidemiology association with delayed paternity reflects factors that may not necessarily be due to increase in age. Epidemiologic association between advanced paternal age and increased offspring risk is particularly well characterized in ASD, most especially, and schizophrenia, but has also been found in other disorders, including congenital heart defect, epilepsy, intellectual disability, among many others. These associations are commonly attributed to age-related de novo mutations, especially the de novo single nucleotide variant. These mutations arise in a paternal germ cells 3 to 4 times more often than they do in maternal germ cells. So paternal germ cells are more they're more prone to this kinds of mutation. In fact, only fathers and fathers will pass down these mutations to majorly their sons or even both sons and daughters or their offspring alike. As men age, the de novo mutations are observed to increase in their offspring. Paternal age-related de novo mutations variants are therefore likely to increase risk for any of the de novo mutations influence disorder. However, the actual magnitude of this risk conferred by such mutations is unknown theoretically, but in practically, but in theory, it's a bit not well defined. So the possibility that the autism spectrum disorder, for example, and schizophrenia association may not be driven primarily by accumulating de novo variants, which received support in a recent study using simulated data that have found that common polygenic risks rather than de novo variants could be most relevant to be observed as cause of ASD or schizophrenia in offspring. However, certain hypotheses are shown that there are certain inherited genetic risk for psychiatric disorder like bipolar or schizophrenia, which may also predict age of childbearing. That is, individuals who carry elevated inherited risk for example, autism spectrum disorder might, on average, have children in later life. The purpose of this study is to estimate the risk for each of the five disorders aforementioned created by paternal age-related de novo mutations in the exomes, the protein code portion of the genomes for the paternal gene. The material and method used, can note that this is a descriptive research, which is a review of over 50 articles collected from online publishing research discussing risks associated with paternal age-related DNVs. And also, we should also note that in the case of CHD, which is congenital heart defect, intellectual disability, and epileptic, epidemiological estimates of paternal age risk are consistent with the magnitude of the effect attributed to the de novo mutations. However, epidemiologic effect for the um, autism spectrum disorders and schizophrenia significantly exceed the risk that can be explained by the de novo mutations alone. Paternal age-related de novo mutation risk model. This risk model was from a statistical model which shows the father of any two given age compares offspring disease risks due to non-synonymous de novo mutation. And this model incorporates two relationships. Number one, the relationship that the general population relation between paternal age and the de novo mutations accumulation, and the case control relationship between de novo mutations and an outcome of interest. If you look at if you look at this slide well, you will notice that there are two bar chart here. One for fathers lower than before the age of 40, and one more than age of 40. And you can see in each of these slides, you can see that many of the schizophrenia, autism spectrum disorder, congenital heart defect, epilepsy, and even intellectual disability, we can see an increase possibility or chances of occurrence in offspring of older or advanced age fathers as compared to age of a lower age, lower than 35, lower than 40. And this using the 1826 SCC control trials in whom data on the paternal age was available, it was discovered that de novo mutation accumulates with advanced parenting age at the rate of 3.1 per year. And so when there's a delayed paternity from mid-20s to mid-40s, for example, a man's offspring would be only about 9% more likely to develop schizophrenia and 20% more likely to develop intellectual disability, consequently to his age-related mutations. So we have been advised that genetic counseling and others can use this data to inform an older or an aged father more than the age of a father at the age of more than 40 years before going into childbirth, because there can be possibility of their offspring developing most especially autism spectrum disorder or schizophrenia. Mental disorders and children neurocognitive ability. A similar amount of attention have been brought or been devoted to the influence of a father's age on the child mental development. The occurrence of this mental disorder, cognitive and developmental abilities, intelligence, and even learning outcomes. Many studies have linked advanced age of the father with behavioral and psychiatric conditions, whereas only a few studies are associated with developmental psychology. The connection is particularly strong in autistic diseases, and like I said earlier, in both autistic diseases and schizophrenia, and in little time, maybe just intellectual disabilities. But while in other cases, a correlation is not well established, like congenital heart defect, which I'll be talking about in the next 2 or 3 slides. However, autism belongs to the neurodevelopmental disorders. Social deficiency, language difficulty, stereotypical behavior, and early development of symptoms are the typical signs of the disease. Advancing paternal age is a significant risk factor for autism in offspring, which has been shown in many studies in recent years. Research has found out that in a cohort of nearly 400,000 individuals of paternal age above 40 years, significantly increase the risk of having a child with autism. Additionally, the father's age can also affect subsequent generation, not just his fourth generation, even the second to third generation. An advanced age father could have a patient or could have offspring or is offering of third generation through the germline can produce an autistic child or autistic offspring. The level of the risk very similar for maternal and paternal grandfather. The grandpaternal age risk estimates were lower than the risk estimate for paternal age, which is true to the hypothesis that the strength of a genetically mediated effect should reflect the genetical relatedness. And the result of one of the most recent reports shows a twofold high probability of autism in offspring of a father age 50 than a father at the age of 30. Moreover, the risk of autism increases with advancing grandparental or paternal age, which may be independent of the age of the mother and the father. It also observed that men above 50 years who have daughters are more likely to have grandchildren with autism. Association between advanced paternal age and other mental disorders like schizophrenia, bipolar disorder, general neurocognitive development have also been observed. Furthermore, the relationship between a higher incidence of birth and the childbirth sequence were also demonstrated. The risk of developing autism was the most significant for firstborn children, and the paternal age effect was also connected with, for example, a higher risk of autism spectrum disorder with intellectual disability. Although different mutations associated with with advanced age and epigenetic alterations are also considered to be the main reason for this phenomenon. However, as I've established before now, that the specific mechanism remain not to be determined. The base for this change may be that the de novo mutations arise in the male germ cells. And furthermore, the knowledge of the quality of the semen decreases with age, and the number of change in a genetical material increases alongside the level of repair. Enzymes are also lower, and DNA fragmentation increases, which also points us to a direction that can I might have led or caused these de novo mutations, which one of the example is schizophrenia, autism spectrum disorder. And just a few words about the paternal age and offspring congenital heart defect. The paternal age, of course, we have made to understand that has been associated with offspring congenital heart defect, which might be caused by the increased mutation in the germline because of accumulation cells, replication, empirical evidence, however, remain inconclusive. We will understand that since the mid of the 20th century, there have been speculation that the paternal age were critically significant for gene mutation. Although the biological mechanism behind the association between paternal age and the offspring congenital heart disease is not clear, but several hypotheses have been proposed. First, increasing mean age is associated with deterioration of semen quality, including volume, sperm concentration, motility, and morphology. Second, there are increasing mutation as increasing germ cell replication error occurs following the advanced paternal age. In addition, older male sperm have increased sperm DNA damage associated with alkaline lipase site or single-stranded DNA breaks. And DNA damage in sperm can be converted to chromosomal aberrations and gene mutation after fertilization. And we're also made to understand that increasing the risk of the developmental defects and genetic disease among offspring. And another hypothesis that was brought out was that exposure to environmental risk factor might be linked to sperm genetic charge or change because of accumulation effect. This was elaborated both in animal model and epidemiological studies. And fourth reason or hypothesis was that for example, young paternal age effect is due to their unhealthy lifestyle among young couples, such as alcohol, smoking, among many other. Furthermore, there has been very limited research on association between paternal age and subtypes of congenital heart defect. Mutation in some of these specific transcription factors I've pointed in a direction. For example, the TBX5 transcription factor genes has been reported, which can induce a secundum atrial septal defect, and a mutation in a CRELD1 encoding a cell ligation molecule can cause atrioventricular septal defect. And it has also been suggested that the TFAP2B gene can cause the Charles syndrome, which is one of the familiar fault of patent ductus arteriosus (PDA). However, all of these studies indicate that, meanwhile, all of these studies indicate that different underlying biological mechanism, among others, are the major prognosis or pathogenesis for different subtypes of congenital heart defect, not necessarily just by the result of paternal age or mutations. Paternal age advance rather than just mutations only. However, a study have suggested that advanced paternal age might be associated with an increased prevalence of a subtype of the congenital heart defects, which is the PDA, patent ductus arteriosus, and not majorly other kinds of congenital heart defect. So although the statistics and mechanism are not significantly and well defined respectively, advanced paternal age and schizophrenia. Quickly, just to make one or two mention, with the help of the diagram just as on the slide, we can see that there's an association between advanced paternal age and increased risk of schizophrenia in offspring, which has been well established over the years. But of course, many of these mechanisms are not well understood. In fact, they are unknown. However, as discussed earlier, the hypothesis is that there's an increased number of de novo mutations in the male germ cells due to advanced age, which might explain the association. It will be interesting for us to know that there is an appealing hypothesis, since it will explain how the incidence of schizophrenia seems to remain stable, although the facility is lower among people with schizophrenia compared to the general population. In a recent study, especially most specifically in 2011, it was hypothesized that delayed fatherhood, meaning advanced age when the first child was born, rather than advancing paternal age per se, contributes to the paternal age effect. This explanation indicates the presence of factors that are associated with both delayed fatherhood and increased risk of schizophrenia in offspring. These factors could be environmental, such as urban culture, paternal genetic traits, impaired social functioning. Furthermore, recent study shows that the paternal age effect is explained by delayed fatherhood rather than advanced paternal age, which is in agreement with other study which points to the fact that offspring to older first-time fathers are at higher risk of developing schizophrenia or non-affective psychotic disorders compared to offspring of older fathers which already have children. Although there is an increase in the number of germ cell division with advanced age that leads to an increased risk of de novo mutations in offspring to aging fathers, but however, there's no strong evidence of association between this mutation and schizophrenia. We can only conclude that there's an hypothesis that the advanced paternal age effect is due to an unknown factor delaying the fatherhood, not necessarily causing an outright psychological effect or mental instability. In conclusion, although there is an increase in number of germ cell division with advanced age that leads to the increased risk of de novo mutation in offspring in aging fathers, several de novo mutations are attributed to factors mostly genetic other than de novo mutations. However, genetic counseling is advised for intended fathers of advanced age in population where the two most prominent de novo mutations that causes autism spectrum disorders, congenital heart disease, neurodevelopmental disability with epilepsy, intellectual disability, schizophrenia are prevalent.